So I'm Christopher Woods. I'm one of the infrastructure leads for the new Isard team looking after the Isard AI supercomputer, which is actually now here in Bristol at this moment being configured. So what is Isard AI? This is an amazing opportunity for Bristol, but also for the UK. Literally only months ago, back in August last year, the government, through the Department of Science, Innovation and Technology, committed over £200 million to build a new AI supercomputer. And we were told in August, can you build an AI supercomputer and have it turned on before the end of March this year? And we went, yes, because we're slightly mad, but we did it. And essentially what you see here is we managed to get this procured. We had it announced for the AI Safety Summit, which took place, hosted in Bletchley Park in November. And then what you see here on the top of the picture with the sort of the car park, the AI supercomputer is being installed in the National Composite Centre, and we have taken their car park. So doing our bit for sustainability by making them take the bus. This is being built from the ground up, from a car park, literally at the beginning of March, to a data centre with a supercomputer by the end of March. This is what the site looked like on Friday last week. So at the back, you have the compound, which is essentially Isard AI Phase 1, and then Isard AI Phase 2 will arrive through this summer, and that's going to be in the sort of the front of the car park here. This sort of will be a big compound. This is inside the compound itself. So actually the data center which holds Isard AI is a modular one. It's a shipping container, and you can see it here on the back of a truck as it was being sort of winched into site in the middle of March. And then phase 1 of Isard AI is the machine, which is, you might be able to see my mouse pointer, hopefully, yeah. It's this machine here just in the back. It's literally the size of a single wardrobe cabinet, and the red and the blue cables here are the water inlet pipes. So there's like tons. This is probably one of the most powerful supercomputers in the UK now, and it is on and it is working. So we hit our deadline of the end of March. But why? What is it for? Well, as I said, it was delivered and funded by the Department of Science, Innovation and Technology directly, and then through UKRI, specifically for AI research in the UK. This is one of the world's only open research AI supercomputers, putting us on a par with, like, you know, big companies and everything else. Specifically, it's for training large language models, large-scale inference, foundational AI research, AI safety, which is very important understanding, machine learning, hybrid AI, etc. Everything that runs on Isard AI must have a strong AI component. And unlike a big supercomputer like an Archer, where you only get to use it if you have a big job, this machine is for everyone. So if you just need one GPU for an hour, you're as welcome to use the machine if you need 1,000 GPUs for a month. So this is like the place to do all AI once we get it going. Why have we spent so much money on AI? Why are we running so quickly to do this? I'm sure we've all heard of ChatGPT, and we've all seen over the last couple of years AI has just rocketed. Artificial intelligence has moved from being a computer science to now being a computational science. This means you can now apply it in your research. If there's a pattern in your data, AI will find the pattern and then make predictions from it. You don't even need to do anything clever. AI will just do that for you. The software you use is now very accessible. It's easy to use in quotes. Obviously, you still have to learn how to do it. And what's cool is as the size of your data and the size of your AI model increases, the capability of your AI model just explodes. It becomes massive. And what you get is this: as you increase the size, you get this emergent capability. Things you didn't expect it to do, suddenly it can do. And as we're sort of going through, we're now understanding how you fit your AI model size to your data size. There's a really nice diagram or movie from Google which illustrates this. When you sort of measure AI model sizes in terms of the number of parameters, when we're kind of working on smaller models, which are sort of 8 billion parameters to kind of just do basic things like language understanding or answering questions, they kind of did that. But as the models grew, you got this emergent behavior. And suddenly it could do summarization, arithmetic, translation, common sense reasoning, semantic parsing, explaining jokes, comprehending reading. And this is why models are getting bigger and bigger and bigger. And what you've seen over the last few years, this is actually a logarithmic plot. It's not even a linear plot. Since 2018, the size of AI models has just exploded. It's gone from sort of the hundreds of millions to billions to now trillions of parameters. And the graph on the right, so it's not great to read, it's basically showing all of the large language models that have come out since 2019. And the ones on the top are the open source ones. This is just a field which is just, it's the fastest field, I think, to appear, which is having a major transformational effect on research. that is what Isard AI is for, is to enable UK researchers to ride this train to make your research better. So how are you going to get ready for it? First, look for the communities that are already building AI models in your field. I can almost guarantee every research field I've looked at, there are communities now applying ML, AI within that field. And if you can't find them, talk in this meeting, but also look at things like Hugging Face. Hugging Faces appear to be the place where people publish their models now and the communities form. Think, what data do you have? What patterns could exist in your data? What questions do you want to ask about your data? Now, I'm afraid you will have to learn a little programming. The language of AI has pretty much become Python, but Python is really easy to learn, and we actually teach Python like anything at the university. Lots of courses which are linked here. Specifically, you want to learn about machine learning, and a good entry point is scikit-learn. But then when you want to go into the really sort of the neural networks, the large language models, those things, those are in PyTorch. And the links here, and you'll get the slides, actually take you to training materials for these. The links also show you how to put it on your own computer or spin up free cloud instances to learn how to do this yourself. And it really is easy to work with once you get into it. We've trained people in a few days to go from nothing to being able to do ML. So it is worth doing. This is all self-guided, which is really nice. Once you've got going on your laptop, you'll soon realize that your model will grow to a size where your laptop will complain because it's too big for your laptop. And that's where Isard AI comes in. It enables you to go from, this is a really good laptop, this is like spec'd out as probably the fastest one you could get for about £3,000. That gives you maybe 8 gigabytes of GPU memory. So its GPU memory is where you fit your model. And it has 468 AI tensor operations per second. So that's how fast it can actually solve it. On Isard AI, your model can become bigger because instead of 8 gigabytes of memory, you've got 384 gigabytes of memory. So your model can be huge. Instead of just running at 466 AI tops, you run at 2,000 tops. So your model will be quicker to train just using one of our processors. But we have 5,000 of them. And if you use all of them together, suddenly you've got nearly hundreds of terabytes of RAM, and you have 10.4 million AI tops. This means that your models can become huge. They will solve, instead of weeks, they'll solve in hours or minutes. It's a resource which I think would be really useful for everybody. So essentially, you'll be able to do more data, larger models, more parameters, faster training, and this is free. You apply for it when DCIT and the Bristol have worked out how you'll apply, and you'll be given this as a kind of a free allocation to work within your research, which will enable us in the UK to really do internationally competitive AI-powered research. So thank you.
My name is Robert Hughes, and I'm here to talk to you about the Bristol Antimicrobial Resistance, or AMR, network. So this is a Bristol-led network similar to the AI Infectious Diseases or Infectious Diseases network at Bristol, but we're specifically trying to bring together researchers from all the different disciplines to focus on antimicrobial resistance. So I'll tell you a little bit about it. Because I'm an engineer and physicist originally by training, I'm going to explain it as if I was explaining it to myself 7 years ago when I started getting involved. So hopefully that's helpful for some of you. This may be common knowledge for a lot of you. But what is AMR? Antimicrobial resistance, or AMR, is basically what it says on the tin. It is the antimicrobials, or these microbes like bacteria, fungi, parasites, viruses, that over time develop resistance to the drugs that we use to try and tackle them. So things like, well, this is a good example really, where you have lots of different pellets here of different antibiotics, and you have a bacteria growing on the surface, and some of those have become resistant to certain antibiotics. This is a massive challenge, and as a society, or as really a civilization, we've only been using antibiotics for less than 100 years. So they were discovered in 1928 and only really started being prescribed in the 1940s. So bacteria hasn't really seen them very much. And so over these past less than 100 years, the increased use of them in various aspects of society, including clinical use, agricultural use, means that they're being exposed, and the generation cycles and evolution cycles of bacteria mean that they can develop resistances very quickly. So this is a massive problem. And as I said, it's not something that can be easily solved by individual disciplines. So in 2016, there was a really good report, the O'Neill report, which basically looked at this as a global problem and how it might be tackled. And they effectively, among other things, proposed that there were a number of different faces that had to be targeted in order to overcome antimicrobial resistance. They included things like public awareness, investment in human capital, drugs, vaccines, alternatives, rapid diagnosis, as well as surveillance and things like that. And these are just some of the recommendations, the ones that are, I think, particularly important or main priorities. But they all have an impact, and there's so many aspects of society in the way that we deal and treat antibiotics or treat people and how we manage them that will affect AMR. So, around the same time as the O'Neill report, Bristol actually put a bid together and put together a community called Bristol Bridge. The idea being that there were many people at the University of Bristol that were all experts in their field and working on areas related to AMR. But this bid was designed to bring together that community and really start tackling those problems more holistically. So Bristol Bridge ran very successfully for many years, and a number of people that are going to be speaking today were involved in that. And the Bristol AMR network is the kind of evolution or the legacy of that community. So myself and a number of volunteers are a part of the committee to manage this community and try to ensure that it stays together and stays coherent and is adaptive to bids and opportunities that are available. In a recent event that we held about 6 months ago, we brought together as many of the community as we could and had discussions about what are the main activities that we as Bristol AMR researchers are looking into. And they included things such as biomedical mechanisms of AMR, detection and diagnosis, animal health, clinical usage, and social and political behaviours and factors. So spanning the whole range of different faculties and disciplines at the University of Bristol. Within that, we also discussed about how these different aspects are all linked, and we essentially came to the conclusion that they are all linked intrinsically, and you can't really have progress in one without supporting progress in the other as well. So this is the kind of research space that Bristol AMR works in, and we work closely with GW4 AMR Alliance as well. Just a little snapshot from the event that we held 6 months ago. Some of you may recognise some of these pictures from the flash talks that were given last year. And credit to all the people that are involved in that. And if you'd like access to any of these, the slides, you can be a part of our network and we can share that with you. But we cover all sorts of things from surveillance, clinical usage, engagement with partners around the world, looking at how different countries deal with antibiotic usage, and as well as nanotechnology, microfluidics, modelling, diagnostics, and then also just interactions with social behaviours as well as a whole range of other activities. So this is the community that we run, and that I wouldn't be doing my job if I didn't plug our event that will be happening in a couple of weeks' time, or at the end of May. So if you'd like to come along and find out about the different research that people in our community do, talk to companies that will be there. So we have companies such as Floretic, a startup that is trying to develop diagnostics to detect bacterial infection much faster. Then please do come along. It'll also be a launch event for an internal impact acceleration call where you can apply to develop a project for maximising impact within AMR. So I think that's me finished. Please use these contact details to get in touch if you'd like to be involved, and yeah, hopefully see some of you at the event. Thanks.
I lead a group
in the health sciences faculty, joint between the medical school and the veterinary school. We do applied health data science and AI, and we have a strong focus on infectious disease and antimicrobial resistance. So Robert just went through a bit of this, but I'm going to start by discussing a little bit our project with Philip Williams, who's a clinician who has a fellowship, an MRC fellowship with us, working with us, and Matthew Avison. And he's interested in antibiotic prescribing and how difficult it can be, and how a lot of it is done empirically, because the clinician may not even know if they have a bacterial infection, let alone if it's resistant to a particular antibiotic. And the diagnostics that are available, it will take, I don't know, you would culture a sample overnight, and you might find the ID of the particular type of bacteria, and then it might take a day or more after that to get antibiotic susceptibility test results. And this is not done for everybody, so this is perhaps done in more serious cases. And the time it takes is a bit of a problem if the patient has sepsis and you need to give them antibiotics urgently. So what we've been looking at is using the history of these antibiotic susceptibility test results to inform the current prescribing. And on the bottom left here, this is the first thing I'm going to show you is almost not AI, because it's a Bayesian model for looking at resistant rate changes over time. They can change, they can be different for all patients than patients in a specific ward. And they're different if you know that the bug is already potentially resistant to your first line antibiotic and you want to switch to another antibiotic. So we make predictions based on the history for the time of the prescription event, and then of developing dashboards that give the clinician an idea, compared to a particular antibiotic, which other antibiotics have more chance to work for this particular patient. And moving on from that, that's essentially a stratified approach. And now we're keenly working on personalized prediction using explainable artificial intelligence. So it's taking that information, but all the history of the patient themselves. And we've been doing this through Health Data Research UK. A huge amount of work by many people has enabled us to link together us and the integrated care board of our region, to link together primary care details and records from 1 million people around here with our laboratory antibiotic susceptibility tests. So this has taken an awful long time to get together. It's very complex data, and now we're at the early stages of developing these AI models. Up here, it shows, for instance, an initial look at the explainability of the AI for particular patients. What's important? What factors are important here in the prediction? Prior resistance is a particularly important one, but you can see there's also correlates with age and other things as well. And then finally, I was going to go on to just briefly introduce you to other things we do. Say we work in the veterinary sphere as well, looking at modelling and prediction that will be helpful in livestock, but also environmental surveillance. I talked before about what you can do after culturing the bacteria. You can pass it through a MALDI mass spectrometer and figure out whether it's E. coli or staph. But that's all you can really do at the moment, and we've got a project looking at extending this to get antimicrobial susceptibility and toxicity direct from that mass spectra, and thus earlier. So that is with Ruth Massey, Matthew Avison and co, and there's a link there. And then for something pretty different, we also work with accelerometry, developing wearable sensors here for goats and sheep in sub-Saharan Africa. Looking at that, the changes in activity that can be correlated and predictive with, say, anthelmintic resistance to worm treatment. And in the bottom right, we have a BBSRC grant at the moment looking at tracking activity and particularly the social behaviors, social interactions between animals that can be, just like humans, the early indicators of impending infectious disease coming up, in this case, mastitis. So I'm going to leave it there. I'm very happy and keen to talk to anyone who has applications as us as data scientists can help you create solutions for. Thank you.
Hello everyone, I must admit I'm a bit of an imposter. I'm not here to talk about infectious disease, I'm talking about non-infectious disease, so please don't kick me out. I want to talk to you a bit about some of the research that I've been up to in COPD. So for anyone who doesn't know, COPD is chronic obstructive pulmonary disease. It's the most common respiratory disease and is the third leading cause of death globally. There's 1.2 million people in the UK who have COPD. And people who have COPD, the symptoms include breathlessness, wheezing, coughing, excessive sputum production, like saliva. And they experience bouts of exacerbations. These are periods of significant worsening of respiratory symptoms. And they can be quite severe. So there's often a high mortality rate, has a serious impact on health-related quality of life, and there are significant health-related costs as well. But we do know early intervention in the course of exacerbations can potentially diminish the severity of the event and also potentially improve health-related quality of life. So for the last 4 years, I've been collaborating with the company Myam Health on the app MyCOPD. It's a digital therapeutic currently prescribed to patients in the NHS. There's over 30,000 users. And their key aim is to try and embed an AI-enabled exacerbation prediction tool within the MyCOPD app. So I'm just going to tell you a bit about how that's been going. So the initial stages of the project were obviously transforming the largest available real-world data set for patient-entered data in COPD into a usable format to apply data analytics and machine learning. And part of that was generating features from this data that could be predictive of exacerbations. And actually, we found one of the most powerful features was the change in the COPD assessment test score. So the COPD assessment test score, or the CAT score, is a measure of how much COPD is affecting a patient's life. It's a clinically validated questionnaire. And from this visualization here in A, about 4 days prior to an exacerbation on day 0, there is a change in CAT score from a stable baseline of 2 points. And that's the mean clinically important difference for the COPD assessment test score, meaning that's indicative of an exacerbation. So we realised we're looking at predicting exacerbations in about a 4-day window, 1 to 4 days before the event. And we've applied various machine learning algorithms, ensemble classifiers, and we found within this patient-entered data, which includes medication usage, CAT scores, symptom scores, how much you're using the app, there is predictive capacity just for how people are using it to manage their day-to-day lives. MyCOPD is mainly used for medication tracking, weather tracking, pulmonary rehabilitation, just self-management. So this kind of modelling and this kind of approach is almost burdenless for the patients. If an AI could be integrated in, it would take very little away from their lives, just how they're using the app normally. After developing some models, we decided it was time to start facing the patients, and we've begun the space studies, sensing predictions and alerts in COPD exacerbations. So in space study 1, we met with some of the MyCOPD users and asked them about AI acceptability. How could this AI be introduced into their use of the app? And there was actually quite a lot of keenness for the integration of this tool, but they felt currently the accuracy was not really usable in their day-to-day lives, and they said there really needs to be an improvement in the current accuracy of the models. And to do that, we realised we need to incorporate some additional sensing. So we conducted our first feasibility study with some MyCOPD patient users and gave them various sensors and conducted interviews to see their perceptions. And we learned a lot from that, which is leading on to the project that we're hoping to initiate soon with about 100 MyCOPD patient users, and we're going to be using 2 key sensors. We found that the Fitbit Inspire 3 is very low burden for patients. In fact, they're almost happy to use the sensor. and it provides overnight heart rate, respiratory rate, and oxygen saturations, which are known to be quite predictive of COPD exacerbations. Also, there's been a recent spin-out company from Cambridge called Tidal Sense, and their new device, N-Tidal Capnometer, which measures CO2 waveforms in exhaled breath. And at the moment, they've said that the CO2 waveforms are pretty good at diagnosing COPD, but there is current lack of evidence whether they can predict exacerbations. However, we're quite hopeful that it could be a very powerful predictor. So incorporating these physiological variables, CO2 waveforms in exhaled breath, and also patient-entered data from the MyCOPD app, we're hopefully going to be generating a very powerful prediction model for exacerbations. And within this study, we're also going to be embedding some patient-facing work, looking at what should an exacerbation warning look like, what kind of information do patients want when they have an exacerbation warning, and also, again, trying to understand the acceptability of AI to patients. How can we translate this information so that the technology is adopted correctly, willingly, and doesn't cause unnecessary concern? But I would very much love to hear anyone's insights about our project, or discuss this with people later. Thank you very much.
I thought I'd bring the primary care perspective. I'm a practicing GP in North Bristol, as well as leading the primary care infection group at the university. And essentially what I wanted to bring to our attention was the variability that infectious disease in the community in primary care brings to the NHS and the challenges that result. So there's a roughly 8-fold average difference between the peak infection rate in usually the winter months for respiratory infections and the trough in the winter, in the summer, I should say. And this presents an enormous challenge to health services and delivery managers. I remember as a junior doctor thinking somebody must have known that my really, really, really busy 24 hours was coming, and that they could have predicted it, and that they could have deployed more staff to make the care safer. Primary care is responsible for 80% of all antibiotics prescribed in the NHS and in health services internationally. So if we're going to improve AMR as a problem, primary care needs to really rise to the challenge. And some more information about what's currently circulating could and has been proven already to influence clinician decision-making when it comes to treatment. Patients, of course, also bring their own perspectives. They want a diagnosis, but for them, the diagnosis, as much as anything, is about what they're going to do subsequently. So is their child going to be able to go to childcare? Are they going to be able to go on a family day out? And do they need an antibiotic? So information about what's currently circulating could potentially not only help health service planners, but also parents and clinicians make better decisions about what to do with their time and where to go. For example, if you know that there's flu circulating, you might choose to do something that's less sociable for your day out with your family. You might choose to go for a lonely walk on the hills rather than go swimming. This is the sort of typical graph that you'll see for pretty much any respiratory infection, be it on a ward in the hospital, be it the clinic activity in primary care, or be it community surveillance. And the point here really is that you can sort of see patterns, right? The key thing is at what point can you predict, a little bit like Henry was saying with COPD, at what point can you predict that this is actually going to become a meaningful outbreak of clinical importance, that we might then want to change our decision making? And I'm no better than the average layperson in terms of my AI knowledge, but what I can see is that these are hugely complex data, for which I suspect AI would be very well suited. One can consider, for example, a range of data sources for where this information could come from. It could come from people's handheld devices in terms of what they're doing. Are they changing their activities? Are they not going to work? Are they not going to school? Are they not engaging in the activities that they were planning to do? The data could come from health services, so the health services already record consultations and requests for consultation, NHS consultations. Of course, secondary care knows exactly what's happening in terms of hospital admissions for infections. Some of that may be simply syndromic data. In other words, it's what your GP or your current hospital doctor tells you you have, which is pretty poor, frankly. I'm a bit ashamed of, as a GP, what I say to my patients. It's a cold, it's flu, it's a viral illness. But nonetheless, we've got those syndromic descriptors. The more we use microbiology, and it's coming more and more in the secondary care sphere, but it's also coming more and more in the primary care sphere, the more we'll have more information about the actual microbes that are circulating at any given time. So there'll be plenty of opportunities to pull these together and start to use them to influence health service planning and then patient behaviour and potentially clinician behaviour as well. Like the others, I'd be delighted to talk to anyone who wants to talk further. I can bring a perspective about what the problems are, what the challenges are, and what I think some of the solutions could be. Thank you.
Hello, so thank you for the opportunity to speak. I won't introduce antimicrobial resistance, others have done an excellent job of that. I have no knowledge of AI, I bring problems rather than solutions. I wanted to talk about two potential applications. So I'm interested in antibiotic discovery. I have access to a potentially novel database of natural products, among which there will be some candidate antibiotics. So my first question is, how can AI potentially identify antibiotic candidates from this distinct area of chemical space? And how can that then reduce the labour associated with isolation and screening of products that turn out to be inactive? This figure is not our work, I pinched it from the literature. This figure over here is partly our work. Yui, who is here, collected many of these images. So my second question is, how can we then identify the mode of action of antibiotic candidates based on analyses of microscopic images? So what we've done here is take a set of comparator agents, compare the response of treated bacteria to our unknown, apply dimensionality reduction to plot these responses in two-dimensional space and identify each of these spots is an individual bacterial cell, that the response to our candidate agent L9 over here differs from the response to the comparator agents. So my second question is, how can we push this approach using AI methodology to improve the resolution of the technique, discriminate known from previously uncharacterized modes of action, and add information by potentially extending into the time domain? So those are my questions. I look forward to potential answers.
Hi, thanks everyone. So my name's Darryl. I study human-restricted bacteria, mainly, how they interplay with our body, and hopefully in some ways trying to stop them causing the nasty diseases they cause within us. So while I've dabbled with neural networks in the past to predict bacterial adhesins, really what I'm interested in is finding solutions to some of the, what at first seem quite simplistic problems, such as you can see here, variable flexible loops of hypervariable amino acid sequences coming together to form the perfect adhesiotope that binds to the human receptor and enables invasive sepsis in some organisms. We've studied different bacterial adhesins and their potential as vaccines, but the same microorganism at different sites in the human body can have very different outcomes. How can we use AI to understand meaningful correlates of protection at different sites in the human body and direct immune strategies and preventative strategies to enable us to eliminate disease, or at least in some instances reduce it? Some things we're also interested in is the uptake of DNA by bacteria and how that enables bacteria to evolve. Some bacterial species really select very specific types of DNA. Some do, and we don't understand why or how. And further, we're moving on to more complex problems. How can we bring together all the multi-omics, both bacterial and human, to understand the role in complex microenvironments such as cancer and degenerative diseases? Thank you.
Hi everyone, I'm Andrew virologist in cellular and molecular medicine. So I work on emerging viral diseases, primarily dengue virus and SARS-CoV-2 and other human pathogenic coronaviruses. And we're very interested in the host response to virus infection with the overarching aims of looking at dysregulated pathways in cells that could be drug targets or lead to prediction of biomarkers for severe disease. So our approaches primarily look at using patient samples from clinical cohorts or infected cells in laboratory, virus infected or uninfected. We subject these to transcriptomics, proteomics, and also lipidomics analysis. We get these large data sets, and then we use those to predict cellular pathways that are disrupted in response to virus infection. However, at the present time, I think we lose a huge amount in the way we're analyzing the data. So I'd like to apply AI approaches to this downstream bioinformatic analysis. And talking to people like Andrew Dowsey before, I know this is quite complex because of a lot of noise in the data, but I'd like to see how we could go further with this. So, thank you.
Hi everybody, thanks for putting on this event. So I'm a lecturer in the vet school, and one aspect of my work involves the study of gastrointestinal disease pathogens. This is done primarily in collaboration with public health agencies like the UKHSA and the CDC, which have really large-scale genomic surveillance programs for specific high-priority pathogens. So in the UK, for instance, a range of different pathogens, when they're detected in clinical care, will be passed to the UKHSA for complete genome sequencing. And then they have like a large-scale overview of the distribution of these diseases within the country, within the UK. What my group does is takes this genomic data, and we have kind of limited information about where they come from surveys that are provisioned to people who have these gastrointestinal diseases, where they travel to in the world, for instance. And we use this metadata alongside the genomic data to try and predict where in the world these pathogens came from. So we can build these kind of hierarchical models where we have information about different continents where these diseases may have arisen, and then the sub-regions within those continents, and then countries within those continents. And we're building this for a range of different gastrointestinal pathogens, looking at how we can use not just machine learning approaches, but explainable AI approaches, or simply how we can integrate these models more effectively with what the outputs are needed by for end users, such as epidemiologists and disease management in public health. So we're sort of focused more on the public health and genomic side of things, but I think areas of interest for collaboration might be how we might generate synthetic data from these genomes, how we might more effectively apply deep learning to genomics data, which is a little bit of a challenge for deep learning at the moment, and how we might approach this using explainable AI, because there's a real need for us to have evidence trails in how we deal with this data so that we can integrate it with public health policy decision making. Thank you very much for listening to me, and I look forward to talking with you later.
I'm Ben. I'm a PhD student in the chemistry department, and I'm in the NMR group. NMR stands for nuclear magnetic resonance, which is a technique that chemists use to help them determine the structure of a molecule that is present in their sample. It's a very similar technology to MRI, except essentially applied to chemistry. So you pop your sample into your NMR spectrometer, and you get out an NMR spectrum. From this, you can figure out the structure of the molecule present in your sample. The issue with this is that analyzing and interpreting these spectra can be quite difficult to do, and generally a skilled or trained chemist is required to do so. So what we are working on and have developed in the NMR group is machine learning approaches for interpreting these spectra and helping the chemist to do this much faster and more efficiently. As ever, I'm interested, therefore, in speaking to people who know about generative models, but also scientists who work in the lab and can help us to understand how we can address the different problems that they come up against. Thanks.
Hello, so I'm Ashish. I'm a third-year PhD candidate at Bristol Veterinary School. So today I will be talking about universal bovine identification. So my interests are in developing deep learning algorithms to non-invasively identify bovines in real time. I'm also interested in developing precision livestock farming tools to enhance animal welfare and disease management. So the method we employ is as follows. We use 3D cameras to measure the surface topology of the animals in the dorsal view. And then we train our deep learning models such that they learn to differentiate the animals. So you can see the colors are individual animals. And then we can use simple clustering algorithms to cluster them based on their identity. The models also learn to focus on certain bony regions, which is quite interesting because experts who deal with body condition scoring also look at these regions. Today, I'm here looking for collaboration in improving our identification method and further deepen our understanding in disease spread patterns. I'm also looking for expertise in health indicators such as body condition scoring and mobility. That's all. Thank you.
So hi, I'm Dan Lawson. I'm going to be talking about pathogen sex, an early warning system for risk. So the idea here is that we're going to be following a database of genomic sequences for a particular pathogen, and we're going to be looking to see where the pathogens look weird. So building a tree out of the genomes, and sometimes on these trees you get these long branches, and one of the reasons that you can get these long branches is because the bacteria have exchanged DNA. They don't look like anything else that's previously existed. So we can measure that with this tool that I'm really shopping for collaborators on. This is chromosome painting, and this allows us to quantify when genes have moved, and from that the goal will be to use AI to predict potential changes in function. And I'd be remiss if I didn't add, I'm also director of JGI, the Data Science Institute, so if you want to talk to me about that, please do later. Thanks.
Hi everyone, I'm Etty. I'm a lecturer in the Statistics Institute in the School of Maths, and my research focuses on infectious disease transmission models and using modern statistics, mostly Bayesian inference, to develop novel methods. I'm mostly interested in looking at how diseases spread through both time and space. Traditional outbreak models have just looked at time only, which I think is a problem. I've got experience in working in real-time modelling, so for outbreaks such as COVID and Ebola, but also I'm kind of interested if there's anybody who's looking at kind of smaller scale disease outbreaks that I'm also interested in that. I mostly focus on low resource settings, so countries which don't have such rich data sources, how can we model these systems where we're in those settings, because that's where the global burden of disease is richest. I'm also passionate about capacity building, so training others, not just in the UK, but worldwide, how to do infectious disease modelling. As an aside, I'm also quite keen to get access to the ALSPAC data, so if anyone here knows how I could do that, please, I'd love to speak to you. Thank you.
Hi there. As you can see, I'm from the computer science department. I know very little about infectious diseases because I'm a computer scientist, so I'm here to talk to you a little bit about the area of my research, causal machine learning. Oops, sorry. There we go. So what is causal machine learning? So it's useful to contrast it with, if you like, regular machine learning. So much of what we do in machine learning is we use training data to build predictive models, which we then use to make predictions about things we don't know about already. So we observe a particular value of something X, and we observe its little x there, and then we make a prediction about Y. So here's an example. We observe that someone has yellow teeth, and from that we predict they're more likely to be a smoker than if they didn't have yellow teeth. Fine, so that's a predictive model. What's going on with causal machine learning? The goal with causal machine learning, as you might expect, is to uncover causal mechanisms. So there we ask a slightly different question. If we were to force a particular variable to take a particular value, what would happen downstream to another variable? So for example, if we took an individual and decided to colour their teeth yellow with a felt tip or whatever, would that increase the probability that they're a smoker? Obviously not, because that's not the way the causality works. So that's what we're trying to do with causal machine learning, to uncover those causal relationships. As you might expect, this is a somewhat controversial line of research with all sorts of interesting philosophical disputes going on between the various protagonists. And some people would argue that without something like a randomized controlled trial or something similar to that, you can't really do it. Other people will say that with appropriate assumptions, even from observational data, you can infer some causal models, and that's what people try to do. So what do these causal models look like? In fact, you saw one earlier in one of the earlier talks where someone had a picture of a Bayesian network. They're directed acyclic graphs, well, most of them are anyway, and where you have arrows from cause to effect. So that's what we're trying to learn, well, that's what I try and learn from data. There's many challenging problems, as you might expect. So often we have important variables which we don't measure in our data, so they're confounding uncovering the causal relationships. That's a real problem. And so it's an area of active research, all sorts of difficult statistical and computational problems. As for my own work, I typically work on combining both data and prior knowledge to try and learn these causal models, because if you've got prior knowledge and don't use it, you're kind of missing out. There we are.
Hello, thanks everybody. So my name is Luca Uggioli. So my sort of expertise is making models of how things move and interact in space and time. And so one specific example is disease transmission. I try to keep with what they're asked. So in specific, I'm interested in connecting, there are big models of epidemiological models that have been used, COVID-19 as an example. So how you actually worry about when things happen at a smaller scale. So I'll give you an example here. So if you're interested, we always got frustrated going to the supermarket when there was COVID-19. No, that's enough. Too many people inside. Why? So then I sort of start thinking about how is it that you can compute or predict that? And so this is sort of the idea. I can make a movie on single slide, but imagine that the fellow in red is moving a bit randomly, and there are the other green fellow that are uninfected, and they move also randomly. How do you compute that? Well, it turns out, I've solved a 100-year-old problem, 3-4 years ago, which allows to actually solve exactly that problem analytically. And so that's a sort of reaction diffusion formalism at a very, so some techniques from the 60s together with what I've done 3-4 years ago, and that allows you to actually do it. So where is AI comes in? Well, I need a computer because the formula that comes out is very big, although it's analytical. It's a big determinant, but you can do it. And in fact, I think it's the first example where R0 is computed exactly, on a model from spatially explicit dependence, okay, or things moving around and so on. So this is in a paper that, if you want to ask me. And so what do I don't know? Well, I don't know a lot of stuff. In particular, I don't know anything about public health, so that's why I'm here. So thanks.
Hi, everyone. I have something a little bit different today. I come from the dark side of industry, so apologies for that. But the good news is that I'm here to help. My background is in infectious disease. I spent my entire career in it, all the way from looking at genomic basis of drug resistance in malaria, all the way to building genomic surveillance infrastructure, pipelines, operations, data products, and resources that the world uses to investigate genomic surveillance at scale. So my background is in population genetics primarily, but then I moved into a much more translational space. And I'm currently the exec VP in the infectious diseases at Airfinity, which is a modeling company that does intelligence around infectious disease transmission, specifically in respiratory pathogens, and working with the UKHSA to understand infection dynamics. But what I'd like to tell you most about is that I work as a Royal Society entrepreneur in residence here. And I think that with my background in infectious diseases, I think particularly with this group of people, I could provide a lot of industry-specific lens to the work that you're doing. And I think that's an incredibly valuable thing as an academic to tap into that kind of experience, because it makes your grants more competitive, it makes your translation to impact more clear and more focused. And so if there's anything I can do, not only in the global health space broadly, but specifically an experience of how you build capacity at scale for these kinds of questions, I'd be very happy to support in any way that I can. I'm here, working 1 day a week at Bristol, but of course, if the EBI had any interest in needing consulting, I'm happy to do that as well. And I see a lot of faces I already recognize from the Bristol crowd here. So yeah, that's it. Thank you.
Hi, thanks very much for having me, and I really enjoyed hearing about a lot of the talks today. And I just want to take a little bit of time to say that I work for the Jean Golden Institute, and I think that we could potentially help with a lot of the projects that have been presented here today. So we are a central hub. We're a university research institute for data science and data-intensive research at the university. And a lot of people don't really know about all the support we can offer. So the first thing I'll pitch is that we offer 8 hours of free support for people with data science queries. People can come to us with a question. It might be a short technical query. It could be advice on applying statistical methods, or it could be proof of concept work for a grant. And typically you'll get help from a second-year PhD student with expertise in data science or AI, also supported by some of our in-house data scientists. And that's the second group I'd like to talk to you about. The best way I can explain the cost of data science support we offer is similar to having access to a pool of postdocs with data science expertise. So if you're writing a grant, or if you have a particular project where you either need a day a week or 4 days a week or a year's time of a data scientist, you can buy out time for someone to support on a project, depending on the resources we have available. Other things we can support with, the JGI organise events, seminars discussing data ethics. We link communities, and for example, this event here, there's communities of health researchers working in AI. If you think you have a particular methodology or people with similar data science interest to you, we could also organize an interest group or an event around that to really build a network. We help deliver and support courses like Christopher Wood's group, like Christopher Wood's courses in R and Python. And in fact, I'll actually go on to this now. From the 3rd to 7th of June, from the 3rd to 7th of June this year, we'll be running Bristol Data Week, where people can have access to free training, but also events on data science and AI at Bristol. And finally, we also offer support for grant development. So if you need advice on how much it would cost to cost data scientist into your bid, if you need help with research planning, for example, what sort of sample size you might need for the analysis you're conducting, or advice on data ethics. And finally, we also fund seed corn projects. So this is small pots of funding available for early career researchers or people later on in their career with a promising idea that has a data science spin on it. And just as an example, earlier for the costed data science support that we were talking about earlier, we have 2 people working in the health domain. I worked with Andy Dowsey in UKHSA on the antimicrobial resistance project, and we have a colleague working with NHS stroke data. So yeah, come find me if you need any help or want to get in touch. Thanks.